Good afternoon. It's a great pleasure to have today Guglielmo Foffani with us. Guglielmo is the research coordinator of CINAC, CINAC, and also he has a part-time position in the Hospital de Parapléjicos in Madrid. He's a biomedical engineer and many other things, but today he's going to talk about his research in the very original hypothesis in the pathogenesis of Parkinson disease. So thank you very much, Guglielmo. It's a pleasure to have you here.
Thank you very much for the invitation. Thank you very much for being here.
No estaba encendido probablemente. Ok, prova, prova, pronto, pronto. Does it work? Yes? Okay, so it's a multidisciplinary team. I'm an engineer. We have other engineers, biologists, physiologists, and we collaborate a lot. There are some clinicians that appear here and there, but most of the clinicians we collaborate with is the team at CIEN CINAC, and we have external collaboration with a number of people, like most research group. So I also recognize the founders at the beginning, so that all the housekeeping is done at the beginning, and it's important to recognize the institution that funds the research. But this story starts in 2015, where we went to the Congress of Movement Disorder in San Diego. At the end of the conferences, I took a flight with a lot of people from the conference back to Madrid through Chicago. And on that flight, I put in place modern shamanic-like technique to enter in an inspired state of consciousness, and I fainted on the plane. And was pretty interesting, the experience with all these neurologists and stuff coming here, all the top people in the field trying to see if I was fine or not. But the interesting part of the story, when the plane landed, I was already vertical and fine, and we had a nice stopover in terms of hours in the airport, and I had this idea, this initial idea of what we call in that moment the Chicago hypothesis, and it took 3 years, and the brainstorming there was with José Obeso, which is the director of my center. It took 3 years to transform that original idea into an actual cortical pathogenic theory of Parkinson disease, which was published in Neuron. And it took 2 years more to find the money to test it. So the initial path, before even starting the research, is a 5-year journey. Of course, was rejected once, the grant, because it's never very easy to arrive to that point. So it took 5 years just to get the money. Of course, you can try to do some stuff in the meantime to get some preliminary data, but is a long path. So now we are in 2025, 10 years from then, and I will kind of share the entire journey of where we are right now. So I will present the theory, tell you about what we did to directly test it in animal models, where we can be a little bit more causal in the approach. We also, I will also show you what we did in patient, which is a little bit more indirect. And what we did in terms of modulating corticostriatal activity toward future disease-modifying therapy. So let's assume the theory is correct. What can we do for therapeutic purposes? So the theory, everybody agrees that Parkinson's disease is associated with loss of a neuron in the substantia nigra part compacta. These are the neurons that project to the striatum and provide dopamine to the striatum. So these are the neurons that die. Dopamine is lost. There is a striatal depletion of dopamine, and this is causally related to the main motor features of the disease. And until here, everybody would agree. Everybody would also agree that at the pathological level, Parkinson's disease is associated not only with the neuronal death. This is the substantia nigra. This is a side that is intact, and this is depleted. The association with these Lewy pathology inclusions, they are very rich of alpha-synuclein that became the fashion protein in Parkinson's disease. But of course, in here there are a lot of proteins. There is more discussion, and what is not very agreeable is the direction of causality. Is the neural death that is causing the Lewy pathology? Is the Lewy pathology that is causing the neural death? Normally, when there are these this causality doubt, there is a bidirectional type of loop, which makes things much more complicated. And all this gained traction like a little bit more than 20 years ago with this paper by Brack, which was the now known Brack staging, is cited like 12,000 times as of today. And the Brack model is a bottom-up theory according to which the alpha-synuclein invades or pathological forms of the protein invades as a kind of a prion-like disease from the periphery, either through the olfactory bulb or through the vagus nerve from the gut. Thank you. And so in the initial stage is outside the brain, enters through the brain, reaches the brainstem and the part that are affected, including the substantia nigra, where that is particularly vulnerable. So the neurons start dying until the point that the disease become clinically manifest. And at latest stages of the disease reaches up to the cortex, and now we are in the very late stages. So this is pretty much how people see overall the disease in the field. There are some issues. Of course, this is a post-mortem based staging, so is naturally cross-sectional. It's not longitudinal. So we all know that you can reach a very wrong conclusion from cross-sectional data. So until you have longitudinal confirmation, this is an hypothesis. It's not hard real stuff. So it's a very nice hypothesis, but as always, there is probably a little more than that. So this fits well. Why it gained traction is that it fit well with, in that moment, Parkinson's disease at the clinical level was started to expand from a primarily motor disorder to a more complex disorder where there are all these non-motor features. Some are late non-motor features, some are early non-motor features. And so all these progression through stages that can be, and location and structure that can be, at least in theory, associated with specific features fitted well with the way of considering the disease. So it kind of helps explaining the complexity of the disease. Here at 0 is diagnosis. You can see some like constipation, REM sleep behavior disorder, hyposmia that can occur early, not necessarily, and other like the classical dyskinesia are the classical long late motor problem due to the dopaminergic therapy, but there are psychosis or dementia, all kind of spectrum of non-motor features that characterize the disease. There is one paradox, which is the starting point of the cortical pathogenic theory. The paradox is that the motor feature start focally. So a little trembling on tremor on a finger or slowness on a leg is very focal. And why this is paradoxical is that the substantia nigra, where supposedly from the bottom-up model you reach the substantia nigra, doesn't have a map of the body to justify a focal onset. There is not a somatotopic representation in the substantia nigra about the body. So how is it possible that a structure that does not have the map has a somatotopic focal onset and even a somatotopic progression, so that adjacent structure become start with the progression in a very clear way? And so we thought, okay, where is the somatotopic? The somatotopic is in the striatum. The projection, they do have somatotopy. And where the somatotopic come from? It comes from the cortex, from the motor and motor-related cortical area. So the queen of somatotopy is the cortex. So the original, let's say the main hypothesis at the higher level is simply that there is a top-down corticostriatal stress factor, somatotopically related, that might just push from the top to the bottom, impinging upon vulnerable terminals in the striatum, and promote this retrograde degeneration of the nigrostriatal cells. This is not technically against Braque bottom-up theory. It's just another thing that instead of going from the bottom to the top, also pushes from the top to the bottom. So it's not alternative. This is important because since I kind of criticized the theory before, we are not proposing the top-down view as an alternative, just something that goes together. And at that point, the concept of retrograde degeneration of nigrostriatal cells was already present. So there is data supporting that retrograde neurodegeneration. We also, and this is the summary of the mechanistic hypothesis. So what may happen at the level of the striatum, how this corticostriatal activity could be a stress factor for dopaminergic cell. So here we have the corticostriatal cell. This is the medium spiny neuron in the striatum, and these are the dopaminergic terminal. So in a very synthesis, this is the synthesis of a lot of papers behind. Most of the alpha-synuclein in the striatum is within the corticostriatal terminal, and it goes out, is excreted to the extracellular space by a calcium-dependent mechanism. Calcium increase means activity. So increasing activity increases the excretion of alpha-synuclein in the extracellular space. The other thing that happened with increasing activity through cholinergic interneurons, we have a modulation of dopamine. So dopamine gets increased in the striatum through this cholinergic interneuron. And this, there is a recent science paper showing that this control actually induces true action potentials in the terminals. So it's not just a little bit of thing. So there is a nice top-down control of dopamine, independently of what happened in the soma, in the substantia nigra, that control the outcome of dopamine in the striatum. So this combination of high level of dopamine and high level of alpha-synuclein in other experiments suggests that is the toxic milieu that enhances the aggregation of the equilibrium, in a sense, of the states of alpha-synuclein toward an aggregated toxic form. And this is the aggregation that would promote the degeneration. So this is what we proposed as a mechanism. Of course, you can see here, if you think about manipulating, which is what we are gonna show you later, there are two ways to try to push this mechanism. One is increase corticostriatal activity. The other is increase alpha-synuclein in corticostriatal cells. Two mechanisms that are purely cortical, that we not mess up at that level. And this is what we try to follow. We also added a generalization of the hypothesis, because if you think what corticostriatal cells do, they also project, this is the cortical spinal pathway that project to the striatum and to all the other structure that are affected in the early Brach stages, receive direct collaterals from the cortical spinal path. So if something is going on at the striatal level, it's probable or possible that similar mechanisms could occur at lower structure. So this effect from the top can affect essentially all the structure that are affected in the Brach staging. So the theory overall has a main hypothesis, a mechanistic hypothesis, a generalization, and if you assume that anything is true, then we start discussing. Okay, then how is the meta hypothesis? Why and how is this cortical activity excessive? And we can think about an overuse mechanism or developmental mechanism. So there is just a variability that I have a little bit more corticostriatal activity than and I can have a greater or lower probability to develop disease, this type of reasoning. Or the simplest is, okay, this is not a primary mechanism, it's a secondary mechanism. So if dopamine start, the depletion start for whatever mechanism, then the loop through the basal ganglia cortical loop will increase cortical activity and will just promote this neurodegeneration in a sort of a positive feedback in a negative sense, but it's a positive feedback loop. And so this is what we had in that paper. Now it is also integrated into in a more recent review. Of course, one of the things that you learn is that theory becomes more real if you talk them more about them. So it's just to repeat the theory all over and over seems to make it more real. You can see the literature all the time. So now that it appears in another review saying, this is a possible theory, all of a sudden it seems more real than it was. But in reality, we need to test it. And what we did to test it was to use animal models, mice. As I told you, the two main manipulation. First one is modulate corticostriatal activity. We did it with chemogenetics, dreads. How many of you are familiar at least with dreads? What a dread is? Silence. So these are, at least with optogenetics. Does optogenetics sound more? Okay, it's the same. So you have a viral vector that induces dreads is designer receptor exclusively activated by designer drugs. So you have a viral vector, then essentially insert in the neurons or the cell that you target a receptor that is sensitive to the synthetic drug that you give. In theory, this is not so, I mean, in practice, this is not as clean as it sounds, but it's worked pretty fine. So essentially, you can have, you can inject CNO, which is the drug, and the CNO depolarize, so hyperexcite the cells that have been infected by the virus. So you can see here, these are the viral infections of the cell. These are layer 5 cortical cells that project to the striatum. We put electrodes in the cortex infected and in the other cortex, and we see this in under anesthesia. You can see the classical up and down states of anesthesia in control. Give CNO on the side that was injected, the cortex starts firing like crazy. And this effect was sufficiently, is sort of a wakefulness-like state. And when we saw that, it was sufficiently striking that we diverted a little bit and published a paper on this local wakefulness under general anesthesia, which actually brought us toward the anesthesia and consciousness in anesthesia type of thinking. But just to say that sometimes when you go on a direction, there are something that you discover on the way, and it's a good thing to try to follow those leads. Going back to our main story, okay, we have this acute effect. So this is the time it takes. So through 1 hour, this is a dermal epidermical injection. Is not intraperitoneal, so it takes a little time, and then it stays for hours. So it doesn't depend very much on the dose. These are all dose over threshold for inducing the effect, and this we were trying to validate the model to use it chronically. So first point, yes, we can induce a unilateral cortical activation of these cells. Then we, in another group of animals, we try to validate the chronic modulation of the corticostriatal activity, and to do that, we needed to sacrifice animals at different time, giving CNO for 5 days a week in a chronic way. We separated animal from which the one that received or not the drug before the perfusion, and to make it short, this is an histological measure of activity. These are early genes. Maybe you know CEFOS is a little bit more known, but this is a EGR1. These are markers of activity in a sense. So from the histology, you can know that the cells that are more dark have been more active in the few hours before the animal was sacrificed. And at the end here, the point is that even in chronic condition, we could still increase the activity of these cells. So we have the acute cortical activation, the chronic cortical activation, and this I missed a point that is important. This is the expression of the virus. Virus was inserted in the cortex, in the striatum. You can see a lot of red on one side, nothing on the other side. Essentially, it's very unilateral model. So we have this acute unilateral activation, chronic unilateral cortical activation, and presumed corticostriatal activation. Technically, we didn't measure the activity of the terminal. We assumed that if the neurons are firing, that information, that activity reaches the terminal. So with that, we have our model of lateralized corticostriatal overactivity. We did something similar with overexpression of alpha-synuclein. Same thing, put the virus now overexpress alpha-synuclein in the target cells. So we target the corticostriatal cell, and we increase either wild type or mutated alpha-synuclein. And you can see again, you might be able to see with the projector, a lot of green here, little green there. So we are increasing expression of alpha-synuclein in the striatum corresponding to the injection, and not in the other. Combining the two things, now we are we are giving the two viruses together, and you can see that they can co-express the dreads and overexpress alpha-synuclein. So at the end, we have this unilateral model of alpha-synuclein and dread overexpression and co-expression. So the conclusion of this part, now we have the models. And we validated the models. And now we went to the main experiment. This is a pretty big experiment. It's actually a sequence of experiments for a total of 115 animals. They essentially received a combination of controls or active viral vectors and the drug that increased the corticostriatal activity that activate the dread during 4 weeks. So for the alpha-synuclein overexpression is a 7-week period. For the activity is a 4-week period, which is long for the experimenters, medium for the animal, short in terms of lifespan, in terms of manipulation. So all this mess of table, just to synthesize, we have animal with control we consider non-active manipulation, independently of what the control was. In all the other are either corticostriatal overactivity, just the dragged, synucleopathy, just alpha-synuclein, or the combination of two. We first studied behavior. Behavior is we just measure the behavior of the animal in an open field for 5 minutes. And what all this does is just a little bit of behavioral overactivity. So the animals are a little bit faster, they move a little bit more, and this is when it's just control and manipulating, where the 4 groups are the different type of manipulation, and they are a little bit asymmetric in the movement, as you would expect from an asymmetric manipulation. Then we went to measure dopaminergic integrity through the TH immunohistochemistry. Nothing. Okay. Maybe something there, nothing very much convincing. So we could not impact on the integrity of dopaminergic cells. We did see a strong astrocytic activation as measured by GFAP with all the manipulations. And this was very, very robust and pretty strong. So this suggests a sort of neuroinflammatory type of situation going on there. You might have noticed there's MPTP there. Who knows what MPTP is? So MPTP is one of the most used toxic as a model of Parkinson's disease. There is a there is an anecdote of how it was discovered. This was discovered through junkies. So all of a sudden, I don't know if it was in the 70s or something, you might know so in San Francisco, all of a sudden there was this Parkinsonian syndrome of people that arrive one over the other, and essentially they cut some drug with these toxins that induced loss of dopaminergic cell and produce a very, very similar Parkinsonian behavior and symptomatology, of course, motor behavior, as Parkinson's disease. So since then, it became a pretty standard way of modeling. The technical thing is the loss of nigrostriatal cells. It does not reproduce the synucleopathy part of the disease. And so what we did in some of these animals was to give one dose of MPTP just to push a little bit the system toward dopaminergic vulnerability. We validated actually this before, but let's go to the point. So we could separate all these animals. First of all, males versus female, same result. It is not sex related, and we also separated the alpha-synuclein and the mutated. Also didn't depend very much on which alpha-synuclein. It did depend, so it was boosted by the MPTP injection. So it sort of is expanded or amplified by a previous or an adjunct vulnerability to the nigrostriatal cells. So what is missing right now from all this stuff to close the paper is we are trying to put all this histological analysis into a pipeline of automatic analysis in the atlas, which is normally how we work in neuroimaging, right? We always have these atlases and we can work in standard space. All I've shown you before was very manual, cutting and going and putting the region of interest. And so we are trying to see this is an actual preliminary data showing the location exactly of the gliosis that we are observing, and it corresponds pretty nicely of where the projection from the cortex arrive. And the synthesis so far is, okay, we have this mild behavioral hyperactivity, which I think is compatible with a little bit of excess of dopamine in a sense. So pushing from the cortex from the top, there is a hyper-dopaminergic effect. Negligible impact on dopaminergic integrity. We are not able to have nigrostriatal neuron die. we do have this robust striatal astrogliosis, which is amplified by nigrostriatal vulnerability. So it's a sort of a first step in supporting this corticostriatal stress can occur. Allora, we didn't formally measure it. You can clearly see it. So if in the cortex there is, the problem is that of course in the cortex is also where we put the injection, so you also do have astrogliosis related to the manipulation. So it becomes a little bit messy. That's why we chose not to manipulate the corticostriata from the striatum, but to be very separate from between where we manipulate and where we measure. So at least we know we are not touching. You know, up there is a little bit... so if we have to update, we need to put the astrocyte in the picture. And the other thing you can think, at the beginning I was thinking a lot in terms of corticostriatal activity. Now that we are manipulating the two things, I think there are these two pathways for corticostriatal stress: activity and alpha-synuclein itself, and they could reinforce each other. So we are now trying to see if corticostriatal overexpression of alpha-synuclein is changing the activity, because at that point it will be, you know, just the alpha-synuclein increases the activity and produces the effect. So this could be absolutely related, and we need to insert the concept of neuroinflammation in the picture, which was implicit before, but now it's very explicit. Let's move to patients. So in patient, we cannot be so mechanistic. We need to get a little bit higher at the main hypothesis level. So it's just this top-down corticostriatal stress. So what we can do, we can think in terms of cortical activity, and to do that, there is this beta oscillations that is the main oscillatory activity that is considered the hallmark of Parkinson disease. This has been studied mostly in subthalamic nucleus and GPI through deep brain stimulation electrode. I started more than 20 years ago doing these recordings during my PhD, and the view, and it's still correct, the view is that these beta oscillation are primarily corticobasal ganglia driven, and even if you are measuring from the STN, a lot of those go through the striatum, so reach the STN through the striatum and can be modulated by dopamine, by the levodopa at striatal level. So what we did was to study this oscillatory activity. This was a part of a work that was not thought in the cortical pathogenic theory framework, but was more methodological. We were trying to understand oscillatory versus non-oscillatory activity. There is a lot of confusion in the field in this concept, a lot of measures that are starting coming out, and we try to make some order. But by the time this is a complex paper, by the time we reach revision, and by the time in the paper we reach figure 7, we obtain this result. So I think that if we had this before being in review, it would have been a higher, we could have targeted a higher journal. But you know, it's a gift of the revision process, so we are happy about it. And you can see on the x-axis is rate of motor progression of the disease, and this is the oscillatory component. So the patient that had more oscillatory activity had, this of course is not longitudinal, so is a posteriori, so is also have to be taken with it's careful, it's not definitive, but they arrived to the point where they were faster than patient that had lower oscillatory activity. Whether the non-oscillatory activity, you consider there is nothing. And in the message is this, right? So faster disease progression if there is more beta activity, which at the STN is a proxy of corticostriatal activity. So at least goes in a good direction. Of course, these are late patients, so it's not early patients. So we cannot go to early patient with DBS because you do not implant patient with brain stimulation. What can we do with in early patients is measure their excitability. You could also do other things. You could do EEG, you could do MEG, you could do neuroimaging. What we are doing is studying cortical excitability through transcranial magnetic stimulation. And this is already 5 years ago. We published this paper in Brain, studying inhibitory mechanism in the cortex. How do you do that? Well, with this TMS stimuli, you essentially excite directly corticospinal cells, which, by the way, leave the collateral in the striatum. So it's pretty much the same cells that we're talking about. And if you give a single test, you measure a twitch, and is a motor evoked potential. If you, before that test, that stimulus, you give a subthreshold stimulus, that subthreshold stimulus is not sufficient to activate the corticospinal tract, but is sufficient to activate the inhibitory circuits in the brain and to inhibit the second stimulus. So the response that you get is smaller than one stimulus alone. By the ratio between the two, you can measure inhibition. And this is a standard measure. This is not anything new. It's a very classical measure. You can see here the motor evoked potential of the test alone and of the paired. So you can see here the difference between these two is the inhibition. In the patient, large sample, 166. And that moment there was like tens of papers which half said cortical disinhibition, cortical inhibition is lost in Parkinson's disease. The other half says not, all with the sample size between 8 and 15. And so we said, okay, let's multiply by 10 the sample size and let's reach something more definitive. You can see a very strong cortical disinhibition. Less and more negative number, more inhibition, more positive or less negative, less inhibition. So a very clear difference, but the most interesting thing that we could separate groups by stages of the disease. These are levodopa naive, very early stage patient. Non-dyskinetic are patients that have been on levodopa treatment but not developing dyskinesia yet, studied off medication, and dyskinetic that have already developed dyskinesia due to levodopa. Also study of medication, no difference in inhibition across disease stages. Of course, cross-sectional. In the very early one, we could actually find, we did have, so here on the x-axis you have the lateralized motor score, clinical motor score. So there are some patients, and these groups are, I don't remember if 10 or 20 per point. These are patients that are absolutely normal on the less affected side. And you can see comparing controls and normal less affected sides, there is already a clear difference in inhibition. So the main view that came out from this study was that this inhibition is something that starts before somehow and sometime before the diagnosis, and is a very early, possibly prodromal feature of the disease. So now we have something affecting the cortex, as a cortex affectation that is very early, and that goes in the direction of disinhibition means more excitability of those circuits. Activity, excitability. Then this paper came out a couple of years ago, kind of contradicting what I just said. And they said, okay, cortical disinhibition drives an association with freezing of gait. Why it contradicts what I said? Because freezing of gait is a late feature, doesn't arrive very early. So we said, okay, we saw the title, what's going on here? We went to the critical figure with the data. And there are two problems. This one is in contradiction with our big study. So these are controls, patient with Parkinson's disease without freezing of gait, patient with Parkinson's disease with freezing of gait that is not responsive to levodopa, or the opposite, responsive to levodopa, patient with freezing of gait, non-responsive to levodopa. You can see that there is no disinhibition, and this is exactly at the same, we tested that 2 millisecond thing, and so we 21, 15, 15, 15, they could not have the result that we have. The second suspicious thing is this very strange box plot. I want to have you try to have that box plot on real data. The point is that these are the Parkinson disease without freezing of gait. If this box plot is not correct, all the paper falls apart, because if this goes a little bit high, then you will have it in both groups. So we said, okay, this is a little bit strange. Let's go and see in our data set. We did have the measure of freezing of gait. It's not as, from the UPDRS, you ask essentially, you see the patient and you say you have it or you don't. So if you see it once, the probability that it has it, but you didn't see, is relatively high. So we included only patients that we had at least 3 clinical measurements, so that the probability of freezing of gait becomes reasonable. And with a group of 32 without and 26 with the freezing of gait, we can see absolutely no difference between the two. So we concluded motor cortex inhibition is not associated with freezing of gait. Little parenthesis, by the way, this is Bayesian statistics. And so if you are losing me all over, the only message that is important and you want to keep from this presentation, throw the classical statistic to the garbage and go to Bayesian. And so I'm saying in a very strong way, because I want you to remember this. I cannot go into that, but just stay with the message. Okay, go back on track. So, okay, freezing of gait doesn't play any role. Of course, we are thinking about early, so we went to the features, non-motor features that are early: constipation, RBD, hyposmia. Let's consider if any of these could play a role in our cortical disinhibition. So if we think about those, there is one that is very special, which is olfactory dysfunction. And it's very special because it's the only one that doesn't seem to progress at all. It's sort of paradoxical. If the patients have it, they have it. If you try to do in the study that go very early, it's there forever. So it starts, nobody knows when it starts, because as early as you go, it's always there. So there are estimations that they could be 10 years, 20 years before it starts. It's just, it seems to be there forever. And this is interesting, the fact that it's not evolving. All the other, like RBD, you can have it before, but you can also have it later. And so it does evolve with the disease. Same thing with constipation, where our cortical disinhibition seems to be pretty stable. So it's a little bit a big jump, but we said, what if there is a correlation between olfactory dysfunction and cortical disinhibition? This was... In the original cortical pathogenic theory, I made a big effort in trying to see, okay, all these olfactory part that is very important in Parkinson's disease doesn't seem to fit with the motor cortex. I couldn't find a link, and so it's not in that original paper. We tried to study a relation experimentally. So what we did was, in a new cohort, study cortical excitability and measure olfaction. This is a very simple measure. You just have these sniff sticks. It takes 5 minutes to run this test, and you have a very nice and validated scores. We measure patients and control, our inhibition and olfactory dysfunction. We get, again, cortical disinhibition. We get the expected olfactory dysfunction in Parkinson's disease. Of course, this was totally expected, a nice separation between the two groups. And whoa, super correlation between this is olfactory dysfunction, this is cortical disinhibition. So more disinhibition, more dysfunction. And the curious thing is that this is both patient and controls. This is only patients. This is only controls. And these are only patients very early, levodopa naive, so it's there always. There is something connecting cortical disinhibition, motor cortex basic circuitry, and olfaction. So you start thinking, say, okay, what's going on here? And of course, we can discuss. The other thing that we did on this data to get a little bit more insight and try to engage a little bit the cognitive abilities of the reviewers with a little bit more sophisticated causal inference type of analysis, which is this mediation analysis, which I don't know if you have heard. So the point here was to try to see if a causal direction. So this is like there is a correlation between a direct effect between the two measures in either direction, and an indirect effect induced by the disease, presence or absence of the disease. And we tested Bayesianly whether this model was more probable than this model. This model is more of an internal mechanism. So this direction from SICI to olfactory, so this inhibition to olfactory dysfunction, you could think that there is something that starts here and goes there, or something's common to both. Whether this will be more if there is an environmental factor that enters through the nose and then goes to the brain and somehow reaches the motor cortex. And so at the end, the probability supported the internal model. Now we can discuss what are the possible connections. There are at least a couple that I think is interesting. One is, and there is a PNAS paper 20 years ago, showing that, of course, when we olfactory function is very much dependent on sniffing. Sniffing is motor. So if in Parkinson disease there is an impairment in the motor part of sniffing that is associated with olfactory dysfunction. So that's one possible direction. The other I like a lot is olfactory receptors. So olfactory receptors, of course, are in the olfactory bulb, but these are and that's why they are called olfactory receptors. But they are olfactory receptor ectopic, so out of place, all over the brain, including substantia nigra, motor cortex, and there is even some evidence of alteration of olfactory reception, of ectopic olfactory reception in the receptors in the frontal cortex in Parkinson disease. So, and there is a very nice possible relationship, also because olfactory receptors are very close to the immunocompatibility complex, which is pretty much related to inflammation. So there is something genetically, it's very far functionally, but genetically these two things might be very, very close. This, of course, is all speculation at this point. And this is in minor revision eye movement disorder since Friday, so it's gonna get out, and it's an interesting result. So all of this, we have this activity that seems to correlate with disease progression. We have this very early, possibly prodromal feature, and this correlation with something that, at least intuitively, might be related to alpha-synuclein. So if there is a mechanism that connects these two, of course, we are not claiming that there is cortical inhibition causing olfactory dysfunction. There has to be some internal mechanism that is up there and that connects these two things. So we need to insert, beta oscillation was kind of obvious in the original formulation. This olfactory dysfunction is a new piece that now requires integration. Last part is, as I said before, let's assume that there is something true in the cortical pathogenic theory. What can we use to exploit it for clinical purposes? And this is the big jump from symptomatic treatment to disease-modifying therapy. The fortunate case of Parkinson disease, it has a lot of symptomatic treatments, and everybody's trying to jump to try to reach a disease-modifying therapy, but it's a hard jump. So what we do is transcranial static magnetic field stimulation. This is our technology, and technology looks a pretty big word because this is really a big magnet on the head. So the original paper was published in 2011. The idea, I remember perfectly, is Antonio Oliviero coming when he came and said, "Okay, what do you think if we put a magnet on the head?" And I said, "Okay, try and let's see what happens." A posteriori, I think it was a brilliant idea, so I inverted a lot of effort in my research to push this technique. It was recognized pretty early, making things as simple as possible. These are the same magnets that you can have on your fridge, neodymium magnet, just much bigger and much stronger, but they are non-invasive, inexpensive, easy to apply, ideal for a sham, for doing research, and fully portable, so you can bring it home. Some nice physical property, the electric field. There is a lot of these electrical stimulation type of things that are affected, or even ultrasound. The big problem is always solve how to go through the tissue and reach the target. The magnetic field is not affected by the tissues, so it's not very focal, but you know exactly where it goes just by the geometry of the magnet that you put. And these are the helmet that we developed. There are several generations of prototypes. From the original ones where we went to this is a nice anecdote. We went to in Toledo, a Toledo pueblo that was the pueblo of leather. And so we went to the best artisan of the leather pueblo, say, look, we want this very, very bad-looking stuff to put in, cylinders in top. He looked at us a little strangely, but he knew we were coming from a reasonable hospital, and we made this first leather-style prototype that was very good for the lab, but has no protection of distance. So was definitely not safe for home use. So these were designed by a designer in Barcelona. There are, it's ergonomic and put distance from the helmet to the outside, so that it doesn't become dangerous, because the magnets are very safe for the brain. But if you put one finger between two magnets, you break your fingers, and that has been experimentally proved, not on purpose, but by a friend, a researcher in Galicia. We have the picture and the imaging evidence that those fingers were broken. So we have to be careful on that side. And this is me in the lab with Claudia and other collaborators. So the step was started we created this startup with Hospital Nacional de Parapléjicos. We published the first paper, first safety study on us, modulation of first in patients with excitability measures, long-term effects, distant effects, first clinical trials, and modulation of corticosteatal activity. I will go very fast through this path. Of course, there are other studies outside. This is essentially the motor part of the evolution. The first paper, so the transition between the first and this one, is the duration of the effects. In the first, so this is the classic experimental paradigm to testing these techniques is motor cortex measuring with TMS before you apply the technique and after. So it's a before-after, very classical paradigm, sham controlled. So in the original, the effect, the magnet was applied for about 10 minutes, and the duration was less than 10 minutes. So a sort of short-term like depression. And in this work, we extended the duration of the application, and by going from 10 or 20 to 30, it jumped to an effect that was already more than 20, 30 minutes. And this is pretty critical in terms of assuming that repetitive applications will sum up, so for clinical applications. So we have this transition from short-term depression to long-term depression. And this categorized the technique between the family of inhibitory and excitatory is an inhibitory technique. There is no way, at least so far, to switch it in the other direction, whether normally with TMS or direct current stimulation, you have protocol that excite and protocol that inhibit. This technique is only inhibitory. With this concept, we went into the first clinical trial in patients with Parkinson's disease with dyskinesia. So we are a little bit off of the cortical pathogenic theory right now. The dyskinesia, the idea was, okay, since it's the output and there is an hyper excitability and hypermotility in the patient, let's reduce the excitability and try to improve the dyskinesia. This was home-based in the sense that we allowed patient to decide whether they preferred to come to the hospital every day or to bring it home. About from 50 patients, this was the total number, one came preferred to come to the hospital because she lived next door and was happy to come and visit. So very good response in terms of feasibility, very good response in terms of side effects. We had a couple of situations with side effects, like for example, two patients had a like panic attack with the magnet, so they don't even want to see the helmet anymore. One was on the real arm, the other was on the sham arm, so unlikely to be related to the stimulation. One fragile patient, I remember, came to the post-evaluation. So sorry I didn't explain. This is 2 weeks, essentially. So it's a pretty short duration treatment with all kind of clinical evaluation before and immediately after. So the patient came at the after session with a periorbital hematoma, probably due to the weight of the magnet, and that resolved, of course. I don't think that she was slammed during the period. Was a very fragile patient. You have to understand that these dyskinetic patients are pretty advanced type of patient, so is not a young type of population. But for the rest, nothing in terms of safety. We didn't get the objective improvement of the dyskinesia. We did get the secondary outcome of the subjective. Sorry, this is the patient global rating of change, and patient that received the real treatment compared to sham scored higher in their subjective opinion about the treatment. So not completely positive trial, but at least a promising concept and a lot of learning from it. We also have a pilot under review in essential tremor, but this is just very short. This is one session and some effect on reducing tremor. I just skip it because we are too off. So the other thing that we did that is relevant for the main story is to move from motor cortex to supplementary motor area. Why we did that? Well, essentially thinking about the theory, not only the motor cortex project to the striatum, the supplementary motor area has a very nice somatotopic representation of the entire body. And the nice thing that it does, it is all in a very small region. When we modulate motor cortex with the magnet, we are in the hand area. So, which is very good for physiology and for research. But if you want to have a clinical application, it makes sense to try to see a region where you are modulating the entire body in a sense. So we move to supplementary motor area. We first, and here the protocol is before, again a before and after treatment, but with resting state fMRI. So subject goes in, subject goes out, stimulation, subject goes in, subject goes out, real versus sham. In the first paper, we saw the local effects of the real compared to sham stimulation, some distant effects in regions that are connected to the stimulated region. And it took us a little bit more effort to actually demonstrate a corticostriatal modulation, which involved all kind of complex methodology development, because functional connectivity is a bad beast. It's something that seems very simple, but is very much affected by local activity. And so if you're interested, I will discuss, but we needed to clean the confounder of the local activity. So with classical functional connectivity, we couldn't see nothing in terms of corticostriatal modulation, which became very, very clear with our model-based functional connectivity. And of course, this and expanded a little bit our possibility of modulation from connected area in the cortex to actually the motor striatum. Was very specific to the motor striatum, so no modulation in non-motor part of the striatum, and so it was a good demonstration that we could modulate corticostriatal activity. And this I'll jump. So what are we doing now? We are actually sending patients at home with the 2 months modulation of is a pilot. This is open, so they know that they are being modulated. We are evaluating cortical excitability and FDG PET MRI protocols. So we are trying to do a nice complete mapping to see if we are able to chronically modulate corticostriatal activity. And this is close to the end. It will take us a while to analyze the data, but a small sample, like 20 patients, and we are now at patient 16, if I remember correctly. So is this jump reasonable or not? To close on a positive side, I want to close on data that are not by us. This is a group in Italy that used our helmet in patients with amyotrophic lateral sclerosis. So in 2021, they published this pilot on 2 patients. This on the y-axis is the clinical scale of ALS. You can see the dramatic degeneration as going down. At point 0, they start with daily, 4 times daily. I mean, it's one side, the other side, one side, the other side session of TMS every day for up to 18 months. And you can see that in this patient, they showed this patient that degeneration, or at least the clinical progression, stopped or was decreased in the other patient. They did a follow-up, the bicentric randomized double-blind placebo-controlled phase 2 trial that was published in Lancet Regional Health Europe a few months ago. And they showed this was 6 months treatment. At 6 months was the primary outcome. At 6 months, patients that received sham were given the option to go to real, and then they followed up until I think 2 years. They failed on the primary outcome at 6 months, but these are the data for the follow-up, and you can see that this is the tracheostomy-free survival probability. So there are patients that are still alive and they're able to breathe on their own, and the patient that started with the real compared to the patient that started with the sham start separating. So of course, this is a relatively small sample, but with ALS is not easy to do these trials with larger samples. It's a very promising result, and they are very active on that side. And there is a nice parallelance between ALS and the cortical pathogenic theory, because in ALS it is much more established, especially of course not through an alpha-synuclein mechanism, but more through glutamatergic excitotoxicity. So the hyperactivity of the motor cortex is contributing to the neurodegeneration. So the logic is the same. The logic is reducing cortical activity. In that sense, is really corticospinal activity and excitability to reduce the overall excitotoxicity. So gives also us a little bit of confidence that the direction also for other degeneration is can be correct. So in there we are already discussing, or other people are discussing, this is ready or not for clinical use, which is an open discussion. But I want to leave you with a current complexity of the cortical pathogenic theory, which already made me realize yesterday I need to do a better figure that integrates all this stuff. I asked ChatGPT to do a figure. Of course, it went in total crazy directions. And just to make sure that I don't pass the wrong message of being a serious scientist, this is the real goal of why I am in science, is an alternative path for being a musician. So in the last couple of years I finally made it. So I put this let's say this dress of scientific outreach in a crazy experimental music project, sent it to FESIT, was funded, and I've been touring for the Caixa forums in Spain and in other places. So that's me with the orchestra and my friend Luca, the violin player, even with the bandoneon, which I'm not really able to play, but I was improvising in a total totally free context, so a lot of fun. And of course, you can also find me writing strange books about tarot and science with improbable parallelisms and stuff like that. Or, as we have been discussing a lot at lunch, writing in actual scientific publication about lucid dreamings and hypnagogic experience or strange stuff that you or others can experience. And with this, thank you very much for your attention and for following me in this 10 years story. Thank you very much.
Fantastic talk. Thank you. Thank you very much. It's amazing how productive was you fainting in that play.
Incredible. It's incredible.
Yeah.
You can try. I mean, and then you tell me 10 years later if it works.
It's so productive. Yeah. Well, some kind of thought that I have listening to you, and we have in neurodegenerative diseases, we have sometimes focal onsets for several etiologies like language or visual, frontal, and with a common, as in sometimes we don't see difference in the proteinopathy. Do you think this mechanism could be also involved in other neurodegenerative diseases with focal origin?
So first of all, we need to refine the concept of focal, because of course there is the brain is topographically focal or somatotopically focal. And I mean, one thing is to say it's more frontal or more occipital or whatever. And another thing is within same structure, there is a somatotopy that is or is not explained. So the focal, that's why I say focal motor, because the problem is the motor focality, which is within a structure that is not somatotopically organized. So having said that, I do think that some of these mechanisms, of course, can be generalized. So in a sense, if you think overexpression of a protein or activity or a mix of the two, it's a general mechanism. At the end, all these neurodegenerative diseases, you change the number of the protein, you mix them up a little bit, you change the probability of the actual place where it starts or evolve, and you transition from one disease to the other. So I think that in the same way that some protein-related mechanism is interesting to try to see. Of course, intracellular, extracellular makes a big difference, but the role played by activity, in a sense, activity from hubs that can touch a lot of area, I think is definitely worth exploring. And I do not want to. Of course, we needed to modulate one protein, and we modulated alpha-synuclein, but I'm not excessively an alpha-synucleocentric person. So I think this is a model. It would be very interesting to overexpress different type of proteins, even in the same circuit. Maybe it ends up being more relevant for Lewy body dementia or something more cortical than Parkinson's disease. That is possible. Maybe with the same modulation, you change the protein, use the ALS protein, and it becomes much more neurodegenerative on other structures. So I think in this type of research, it's very good to be open and see the cross fertility for other, at least for other diseases and for other mechanisms. So yeah, we can speculate, and it will be interesting. Of course, it's years of work then to try to put in place any of the idea that we can put on a piece of paper with a beer. But is I think so. The most logical extension is Lewy body dementia. Right, so now we are modulating motor cortex. We could do exactly the same, moving outside the motor cortex. Motor cortex is particularly non-vulnerable in terms of neurodegeneration, which make it on one sense, very interesting, because if he's non-vulnerable, doesn't mean that he's not involved in the process. It's just that is the last dying, but it could be doing disaster everywhere else. So I find it very, very interesting in that sense. But of course, we could do the same in more vulnerable, in a sense, cortical areas that are more prone to Lewy body expression and stuff, and probably the effect in a neurodegenerative sense could become more cortical and yeah.
And yes, one clinical comment before I pass the mic. A detail that you didn't mention is that it's, yes, I mean, Parkinson's disease is normally asymmetric, and normally it's the dominant side, which sort of.
So it's okay. Yes, it is definitely in the paper I discuss in the original theory. There is this laterality. It's not huge, so it's 60-40. Yes, it is. Is it demonstrated? It's 60-40. There is a higher probability of starting on the side where there is dominance. Of course, dominance is not only defined on the hand, but dominance is a more complex concept, because there is hand dominant, leg dominant, eye dominance, nose dominance, and this is much less studied. I think that if it was studied more completely, probably the association could be stronger. And of course, when you start thinking in dominance, well, cortical architecture seems to play a role. So it's a good indirect argument that the cortex might be involved in the direction of the neurodegeneration. So it's definitely a good point and a good argument in favor of the overall theory.
Yeah, you already mentioned that that this theory is not like competing with the traditional Braque style progression of pathology and neuronal death. So I wonder whether you see that as an, right, well, it can be in addition to this standard Braque progression model, but I wonder whether you see it like as a general mechanism in Parkinson's, or whether it may affect some specific types of Parkinson, like subtypes. You already mentioned dementia with Lewy bodies. Maybe it's more relevant for some or the other. There's this brain first, body first hypothesis about subtypes.
I think it's a very important point, and I do not have, of course, a clean answer. I think the body first, brain first, in my view, is an evolution of Braque. So it's still very bottom up. It's just instead of saying it comes at the same time from the nose and the gut, it is an either. So it's again, it's very nice dichotomization, because there are clearly two paths, but it's still a very bottom up type of approach. So in this sense, in which one this might play, I don't know. It is possible, yes. It is very possible. We could not, from our data, for example, on the TMS data, to separate two populations that clearly have or do not have the cortical disinhibition. It is true that we have a bias of inclusion, because with TMS you cannot study tremor dominant Parkinson's disease. So now already a big piece of the clinical dichotomy is gone. So I cannot exclude that can be something of that. Of course, we are going in the direction in trying to map a little bit better all these symptoms. So, for example, even in the body first view, if you have RBD or something, or constipation will be more in this body first concept, could be very much affected by the cortex, so it's not in competition. It will be very interesting to see if this RBD starting type of Parkinson's disease has or has not a clear cortical disinhibition. Preliminary data by others published very small sample size again, show that there can be already a cortical disinhibition, but I think they are not definitive. I would definitely like to run a bigger study, but it's not so easy to get those patients. So yes, I do think is a very right direction to go, and we can go in that direction in patients because the variability of the measure that we have is very high, and at least part of that variability has to be related to biological variability on the disease. So in which one will end up to be relevant to the cortical pathogenic theory?
So
Who knows?
you mentioned it as a possibly prodromal feature. That's why it would be interesting. Well, that's why it's so interesting to study it with olfactory dysfunction or IRBD and these things. What about genetic cases?
So we are trying also that at CINAC, at least the early patients are being tested for genetics. Of course, that's why we were discussing. I was interested about the genetic profile. I think there are two directions. The most obvious is genes that are related to Parkinson, and of course you end up with very small sample size. So to get a decent amount of specific mutations, you need a huge amount of data, and so is a lot of TMS to try to do on that. But it's also possible. I mean, if you expand to more normal-like type of things. So it doesn't have to be exactly. It's not that the cortical effect need to be associated with necessarily with a Parkinson-related gene.
You saw the correlation even in controls.
The correlation with olfactory function was also in control. So there might be some very general mechanism that unite those two things, and then they, in the presence of Parkinson, they become more negative in a pathological sense.
That would be my next and last question. Where does this cortical disinhibition come from?
So that was in a sense was what I called the meta hypothesis. So I think that there are, if we assume that it comes before the disease, is either it's either a very early part of the same disease process, so that it develops like olfaction, right? The same logic for olfaction. The olfaction problem is a trait, or is a very early disease feature. This is the discussion. So the same thing, the cortical dysfunction that can assume that it's doing something negative is a trait. So it's a developmental thing, or it is something that is a very early starting point of the disease for some mechanism that we don't know. This is, I think, is where we can discuss about it. Of course, it's not easy to go and demonstrate what it is.
I mean, we were discussing genetics, but of course, for genetics you need big numbers. But it would be a way also to try to. We were discussing Mendelian randomization, so you can try to look at the risk genes that are related to Parkinson disease and see whether they are also related to cortical disinhibition, to see if there is a common background, genetic background that are related to these things that could be present. If they are common, they could be present in controls perfectly. But for that, you need, I guess, big numbers with cortical disinhibition being assessed.
So the good thing of the correlation with olfaction is that there is much more data on olfaction. So if we are able to understand a little bit more mechanistically this correlation, then olfaction become and it's and there is a lot of I did some searching in the literature.
But olfaction and Parkinson are already linked.
Exactly. So at the end, of course, it is always indirect, but you can kind of advance in the direction, and it becomes very, very. Of course, in big you're used to work with big cohorts with projection, right? Of course, if you had, it's probably not very feasible to do TMS in this sense. That's why we need some other, but just the corticostriatal
do you have
activity.
a proxy for cortical disinhibition, something that you can use more, scale more in?
of action,
action.
but really doesn't make any sense. So that's why we need to have a better, you know, one of the things that we would like to do is, of course, even in this in this group that we are modulating with the 2 months, we are having a more complete set of neuroimaging and inhibitory things. So we are also playing a little bit with other measures with TMS that are a little bit more complex and try to see which one can be, you know, if there was an equivalent on neuroimaging that correlate with cortical disinhibition, it would be nice. Some attempts have been done in literature with spectroscopy, but GABA spectroscopy does not seem to correlate with cortical disinhibition in the same sense. So is you know, then we ended up not understanding what we are measuring, rather than trying to, even if they have the same name, they are both GABAergic related, but probably capturing different aspects of GABA physiology. So we need something simple to scale. Yeah.
In your theory, this cortical stress might retroproject to the to the striatum, but we know from imaging that there is a pattern in striatum, that depletion that is heterogeneous but pretty consistent. It's not like it will appear in the middle of the striatum. Generally, it will always start from the tail and progress further. So we should expect that similar, some sort of heterogeneity, but probably will not explain that sometimes the symptoms start in the right hand and other time in the toe, in the feet. That would be much more complex to explain. So I wonder how this more or less consistent that depletion would fit with the theory.
So I think there are several issues there. First, so let me see different angles. First angle is the focality could be very well a starting point that is conceptual, and then is not so important, because it's really, you know, it's to say, okay, why it comes from, but it's really more general, and the focality is just a threshold-based type of thing, so that by stochastically process, you end up that you are closer to the clinical threshold on some point than the other, and by simply by spatial vicinity, you have the somatotopic progression. And so the cortex could just be a overall pushing in that direction that helps into the X process. So it's not easy to demonstrate the focality in a totally clean sense. The other opposite is we do not have neuroimaging real resolution to actually study the focality at the neuroimaging level. So it could be on the other side, right? So you say, okay, it looks like the pre-stochastic and general neurodegeneration. In reality, it might be more specific, that it looks on a FDG on a DOPA PET. Than what it actually is. So at the end, you can see the angle for.
I would buy the second point more, because the first one seems to fit very well with your theory.
Okay, so if it's you going with the theory, it's better.
If I were you, I would.
Of course. But you know, I try. Yeah. But I try to be fair. So you know, to explore. Even I think that the stochastic thing was even in the original paper. You know, nothing is really black or white. So it's just directions that with higher or less probability. So we are playing with probabilities.
Have you found in your studies with the stimulation any effect on cognitive function?
On cognitive function.
Allora, a parallel question: if you're going to act on the cognition, where would you like you place the stimulation?
So with the motor and SMA stimulation, we did not even measure possible cognitive effect. There is a study in which I was involved. depending what you consider cognitive. But for example, if you go on visual cortex, there was clearly an impact on more visual spatial type of task. So there is not very clear if it's the cognitive or the visual. It's probably more on the visual part. And of course, if you go in area where you expect that inhibited area will have a cognitive impact, you can have cognitive effects. The point is that you have to remember you're inhibiting. Of course, also the concept of inhibiting is a simplification, because inhibiting is based on a decreased motor evoked potential in the motor cortex. So, for example, in the visual cortex, that effect was mediated by an increase of alpha. So occipital alpha goes well with the idea of decreasing the activity of the cortex, so the resting alpha increases. Something similar happened in somatosensory cortex. We have a study on somatosensory cortex where the behavioral effect is the opposite. So in the occipital alpha was associated with impairment of the task, whether in the somatosensory cortex with an improvement of the task. But that's not related to the technique, is related to the relationship between the ongoing alpha and the task. So the alpha was modulated in the same direction. In one task, there was a positive correlation between alpha and the outcome, and the other was negative. So at the end, you end up in different places. Thinking about other target, I don't think there is a lot, which means a lot can be done. So it's and I've never worked on heavily on cognition. So I would not be. I can search where to go depending on what type of task, with the limitation of the inhibitory protocol.
So you have not any suggestion to apply this technique of stimulation to Alzheimer's disease?
So I think.
The focality.
So I think. That if I went toward Alzheimer, my thinking is more in terms of what it does, because this is not studied, to non-neural population. So, for example, we do not know what it does to glia. We do not know what it does to the blood-brain barrier. We do not know really what it does at many other levels. So in more than thinking in a classical functional way, I think even in the ELS, I'm not totally sure that the effect is mediated by neural activity. It could be very much mediated by all the other cells that are around. So I think the direction toward a diffuse disease is not. I wouldn't try to do a symptomatic improvement of a specific symptom, but more try to see if there is a more fundamental relationship between the magnetic field and the inflammation. For example, magnetic fields are very well known to impact inflammation in peripheral tissue. So they're used for knee injury or those type of things. Magnetotherapy is used in physiotherapy. So it is not very well studied what happened in terms of neuroinflammation, the exposure to the static magnetic field in the brain. So that's, I think, it's a more promising direction for a disease like Alzheimer. I also do not know how much what is the role of neuroinflammation in Alzheimer, but in this disease is always a mess that you never know who is the egg and who is the chicken. So if you can decrease the neuroinflammation, is always a good thing.
Quite a powerful magnet in the basement. We could use.
So that's a
for magnet.
Let's just leave them there. Not doing anything. Just leave them there and wait here for an hour.
Make a bit longer the protocol. That's already.
Yeah, it's and I'm not sure if there is because with so much imaging done on people, if there is anybody that studied the actual on biomarkers on stuff
It's
about
been done with CT, with CT scanner, because there are some reports that individuals that repeat the CT scan improve a little bit, And actually, there are studies going on to try to test the effect of radiotherapy at low levels, because it's also may modulate inflammation.
Yeah, I think it's the same logic with the magnetic field. It should decrease the inflammation.
Yeah.
So it's probably good to go in the scanner. This is good for if you want to get volunteers. You know, you have to tell them.
Yeah, exactly. Well, thank you very much. It was a great pleasure to have you here at the discussion. Thank you.
Thank you.